The overarching theme for today's lecture is cellular reproduction or cell division. The ability to reproduce distinguishes living organisms from non-living things. Cell division is defined as a process in which a cell, that is called the parent cell, divides into two or more cells that are referred to as daughter cells. Now, there are many functions associated with cell division. Those functions are listed here for you on this slide. Again, one of the functions of cell division is reproduction, to reproduce. The other functions include growth and development of an organism, but also can function in a mechanism of repair. Now, briefly, when we talk about cell division in prokaryotes, this is carried out by a mechanism known as binary fission. I'm not going to cover that in this particular lecture. However, your textbook does go over binary fission in prokaryotes, and I do suggest you review that. In eukaryotes, there are two distinct mechanisms of nuclear division known as mitosis and meiosis, and both of those, if they are followed by a mechanism known as cytokinesis, will then result in cell division. A really nice summary of both mitosis and meiosis at the organismal level is seen on this particular figure. If you look at the dog on the left-hand side of the figure, this is an adult dog, and if this dog is going to reproduce, there are cells that need to undergo the process of meiosis. Meiosis is a mechanism where there's a reduction in the genetic material by 1/2, and we will look at this in a lot more detail in today's lecture. But the process of meiosis, followed by cell division, results in either the production of egg or sperm, also referred to as gametes. Gametes are then utilized in fertilization to produce a new and unique, distinct individual. That single fertilized egg will need to undergo the process of mitosis, followed by cell division, to produce a fully grown adult dog. So this process is a circular process, and you can see both mechanisms are really important for the survival of the individual or the species. In the mechanism of mitosis, there is a distribution of identical genetic material into two daughter cells. And again, we will look at that mechanism in more detail in today's lecture. Now, before we jump into cellular division or cellular reproduction, let's actually take a few steps back and look at organismal mechanisms of reproduction. And that's important because they also tie into the cellular mechanisms of reproduction. So one broad mechanism for reproduction is known as asexual reproduction. In asexual reproduction, there is no fertilization of gametes that takes place, and this is a mechanism where organisms produce genetically identical organisms that can stand on their own. This is common in single-celled organisms, such as the amoeba, like you see on the slide on the left-hand side, but there are also many multicellular organisms that can reproduce asexually. And again, asexually just means that there's no fertilization of gametes that takes place. There is production of new individuals, but those new individuals are genetically identical to the original individuals. Sea stars, for example, one of the arms can break off and begin to develop into an entirely new individual. Again, genetically identical to the first individual, but a distinct individual nonetheless. Plants are really well known for doing this. The African violet, like you see in this particular figure, can produce genetically identical individuals that are distinct plants. Now, the other well-known mechanism of organismal reproduction is referred to as sexual reproduction. And in sexual reproduction, simply what we have from the biological sense is a fertilization of two distinct gametes producing offspring that are genetically distinct from the individuals that are contributing the genetic information. So we revisit the slide that we saw on the first slide in the lecture, where you have an adult dog. If the adult dog is going to go ahead and reproduce, Specific cells need to be formed that are known as gametes, and the way that these cells are formed is through a process known as meiosis, followed by cytokinesis, producing the gametes that will be utilized in sexual reproduction. Once you have gametes formed, you can have a fusion of those gametes take place in a fertilization event, producing a fertilized egg, and like I mentioned in the first slide, that fertilized egg will then begin to divide via the mechanism of nuclear division known as mitosis, followed by cytokinesis, to produce the cells that make up the individual organism. And again, you can see this is a cyclic process. But the important point here is the sexual reproduction mechanism that I've highlighted with the definition, again, requires fertilization by the fusion of gametes producing offspring that are genetically distinct from both of the parents that contributed genetic information. Now, this is important because as we begin to talk about cellular reproduction, we're going to look at both of these mechanisms of nuclear division, mitosis and meiosis, in quite a bit of detail. And it's important to understand what the processes result in and why they are important for the organism as a whole. As we continue to delve a little deeper into cellular reproduction and looking specifically at the mechanisms of nuclear division known as mitosis and meiosis, what we also need to do is take a step back and discuss chromosome structure in a little bit of detail. Now, chromosomes are organized structures that contain DNA and proteins, again, DNA being the genetic material of the cell. When we focus on the nuclear division mechanisms of mitosis and meiosis, the most important thing that is taking place during that process is the division of nuclear material or genetic material. So we need to understand chromosome structure to then be able to understand what is happening during the partitioning of that genetic material in either mitosis and meiosis. So again, a chromosome is a distinct organized structure that contains the genetic material, DNA, as well as proteins. Eukaryotic chromosomes are distinct structures that contain the genetic material of the cell and the organism. But there is a lot of detail associated with chromosomes and how they are formed and their structural organization. If you look at the top of the slide, you can see the DNA double helix, which is in fact the genetic material of the cell. And at this particular point, I usually refer to that as naked DNA, meaning it's just DNA with no association of any other type of molecule. When we learn about DNA, this is the structure or the form that we typically learn about. However, in the eukaryotic nucleus, DNA is not found in this naked state. It is found in complex organization with proteins, a family of proteins that are known as histones. And you can see that on the slide as well with those little purple spheres. So histones are a family of proteins that complex with DNA to begin to form the complex chromosome structure. The DNA is organized in such a way to be able to compact a large amount of genetic material into the nucleus of the cell, and this is why chromosome structure is really, really important. When you look at the breakdown of the typical chromosome, we can talk about some characteristics associated with chromosome structure. So on the slide, you also see the term nucleosomes. Nucleosome is singular, and this is the first level of organization in chromosome structure. It is how the DNA and the histones come together. So the histones, again, are a family of proteins, and in this diagram are just represented by a circle. What happens in the organization of the DNA is that the DNA is wrapped around the histones to help compact the DNA. So that's what you see in the center of the slide. Now, this is also sometimes referred to as beads on a string, because if you look at it under an electron microscope, that's exactly what it looks like. However, that's just the first level of organization for chromosome structure. There are several complex layers of organization, basically where these beads on a string are wound into looped domains to eventually form the complex chromosome structure. Another very important feature about chromosomes and chromosome structure is highlighted on this particular slide. When you look at the figure on the right-hand side, this is an electron micrograph image of a chromosome. And this characteristic X-like structure is what we typically think of when we think of a chromosome. However, what I want to highlight for you is that that characteristic X-like structure does not represent a chromosome in the majority of the time of the cell life or cycle. The chromosome that you see in this particular figure with a characteristic X-like structure is what's referred to as a duplicated chromosome. So in that same figure, in the cartoon version on the left at the very top, you see what would be the representation of a single chromosome, sort of like that single bar at the very top. What happens in preparation for cell division, whether it's mitosis or meiosis, is all of the genetic material is copied, replicated, or duplicated. When that happens, you have 2 copies of each chromosome present inside the nucleus of the cell. So right in the center of that particular figure, you can see what's referred to as the duplicated chromosome. So the electron micrograph image on the right-hand side is also a representation of a duplicated chromosome. What is going to happen throughout the process of either mitosis or meiosis is that those 2 copies are going to separate from one another. Now, other terms that you see on this slide, one, for example, is the word centromere. The centromere is a highly condensed portion of the chromosome right in the middle that's sort of pinched in where the X-like structure looks like it's pinched in. This is where the 2 duplicated copies of the chromosome are attached physically to one another. So the centromere, again, is a highly condensed portion of the chromosome where the 2 duplicated chromosomes attach to one another. You also see on here the term sister chromatids. Sister chromatids is what we refer to as the 2 copies of the chromosome that are produced after chromosome duplication or replication. So in the characteristic X-like structure, if you went all the way from the top to bottom on the left-hand side, that might represent the original chromosome, and on the right-hand side from top to bottom would be the duplicated chromosome. In this case, you now have 2 sister chromatids, which are the duplicated copies of the chromosome, collectively referred to as sister chromatids. Before we jump into talking about mitosis, I want to discuss what's known as the cell cycle. The cell cycle is an ordered sequence of events of a eukaryotic cell from its origin in cell division from a parent cell until it divides into 2 cells. This can also be referred to as the life cycle of the cell, from when it arose from a cell division event until it divides and produces daughter cells as well. When we talk about the cell cycle in eukaryotes, it is divided into 2 main phases. There is what's known as interphase, and interphase is when normal cellular growth occurs, accumulation of nutrients occurs, and 90% of the cell cycle is spent in interphase. So this is sort of the everyday life of the cell is what we would consider interphase. Again, 90% of the cell cycle, 90% of the time, is spent in interphase. The other 10% of the time would be referred to as mitosis, and this would be the cell division event, the nuclear division event, followed by cytokinesis that would result in cell division. This is going to be when the cell splits into 2 genetically identical daughter cells. So when you look at this particular figure, you can see the cyclic representation of the cell cycle from the time that the cell arises from a cell division event. In the greenish color, you have the interphase portion of the cell cycle, and then that small 10% portion of the pie in orange represents the mitotic phase, the M phase, or mitosis. So interphase is when you would have the normal life cycle of a given cell, the normal everyday life of a cell taking place. We can subdivide interphase into subphases as well. These phases are known as G1, S, and G2. The G in G1 and G2 stands for gap, and that's because originally it wasn't clear what was happening during this portion of interphase, and then it just seemed to be a gap overall. We now know that there are important processes that are taking place in both G1 and G2, but let's look at them in a little more detail now. So G1, or the first gap, is when typical metabolism and growth is going to take place. Now remember, the cell cycle begins from the time a cell arises from a cell division event. That cell is actually going to be smaller typically than the normal size of the cell that is going to enter into a division event. So the cell does need to grow, and again, normal metabolic activity is going to take place during the G1 phase. At the very end of the G1 phase, there is a cell signal that causes the cell to enter or begin the process of entering into mitosis. Now, this is still interphase, but there is a signal that tells the cell that the conditions are favorable to go ahead and begin the process of preparing for mitosis. The next subphase in interphase is the S phase, and the S stands for synthesis. This is specifically the synthesis of DNA. As I mentioned in a previous slide, in order for either mitosis or meiosis to occur, all of the genetic material has to be copied, duplicated, or replicated. So during the S phase is when the synthesis of DNA takes place. DNA duplication, replication is taking place during the S phase of interphase. In G2, there are some additional mechanisms that takes place. The cell begins to form chromosome structure, although the distinct chromosome structure that we're going to talk about really is not visible under the light microscope until we enter into mitosis, and I'll highlight that when we get to that portion of the lecture. But during the G2 phase, the cell is completing preparation for cell and nuclear division to take place. As you might expect, there is complex molecular mechanisms involved in the regulation of the cell cycle. There are both internal and external cues that need to be recognized and met in order for passing of one phase into the next phase during the cell cycle. During G1, specifically, there is a checkpoint known as the start checkpoint. What happens here during G1 is that cell growth is checked. Is the cell really big enough to continue on with an eventual cell division event? Is the environment favorable to allow for a cell division event? So for example, if there are not enough nutrients around, it doesn't make sense for the cell to divide to produce two resulting daughter cells that are also going to take up nutrients if those nutrients are scarce. So is the environment favorable? Yes, then the checkpoint can be passed and the next phase can be entered. There is also a checkpoint in G2. In G2, this is just before entering into mitosis. Again, cell growth is checked. Is the cell, again, big enough to continue on this process? Is the environmental conditions okay for reproducing the cell? And more importantly, has all of the DNA completed the replication process? If that process has not been completed during the S phase, there is no sense to continue on with mitosis, because what's going to happen during mitosis is the partitioning of the genetic material. And if all of the genetic material has not been copied and duplicated correctly, then the partitioning of that material cannot take place properly and correctly. Not really the topic of today's lecture, but just interestingly, cancer cells are cells that divide excessively and then eventually invade other tissues in the individual. Cancer cells are cells that typically have escaped these control mechanisms that allow for checking for the environmental conditions, checking to make sure the cell is ready to divide, and so on. And so, again, many types of cancer have escaped these checkpoints to continue dividing excessively. This slide and the next slide are going to look at the sub-phases of mitosis in much more detail. But before we do that, let's go back and review what's happening during interphase, specifically during the S phase of interphase. This is when DNA synthesis occurs, DNA replication, DNA duplication, the copying of all the genetic material must take place before mitosis can actually begin. So that does in fact happen during the S phase of interphase, and that's a really important step. As I mentioned in the previous slide, there are checkpoints that need to be met in order for the cell to proceed. If all of those checkpoints have been checked and met, then the cell can proceed into mitosis. So the first phase or subphase of mitosis is known as prophase. Now, in this figure and in the following slide, I have both the cartoon image of a cell as well as a series of slides or images of cells that have been stained with fluorescent dyes that specifically bind to components of the cell. In those images, those are the ones on the bottom half of the slide. In the red, you see a red fluorescent dye that binds to the cytoskeletal component known as actin. The green specifically binds to a cytoskeleton component known as microtubules, and the bluish purple color specifically binds to DNA. So you can highlight the different components that are really important during the mitosis process. I also mentioned previously that the most important thing that happens during mitosis is that this is a nuclear division event, that the genetic material is going to be partitioned so that the resulting cells that are produced at the end of mitosis followed by cell division have complete sets of genetic information or genetic material present. So what you see on the cartoon version of the slide is highlighting that genetic material and what's happening to chromosomes during the process. All right, so back to prophase. In prophase, what we have are some distinct features or characteristics taking place. The chromosomes begin to condense or coil up so that under the light microscope we can begin to see that chromosome structure. So chromosomes begin to condense. We begin to see mitotic spindle fibers form. Mitotic spindle fibers are made up of microtubules, and they begin to organize and form during prophase. And that's what you see in the center of your slide. On the right-hand side, you have a phase known as Metaphase. Now, it's not listed on the slide, but this is definitely a phase known as Metaphase. Metaphase, you begin to see those chromosomes completely condense, so we see that characteristic X-like structure. You can also see that the microtubules have attached to the centromeres that highly condensed portion of the chromosome that holds the sister chromatids together. So these are characteristic features of Metaphase. The next phase or stage is known as Metaphase, and you see that in the left-hand side of the slide. In Metaphase, the characteristic feature of what's happening is that the chromosomes align along a central axis of the cell. How do the chromosomes align? Well, those microtubules that have attached themselves to the centromeres essentially push and pull the chromosomes so they are lined up along that central axis. So again, in the nice cartoon version, you could draw a line straight down where those chromosomes are along this central axis of the cell. in the actual cell that's been highlighted with the fluorescent dyes, you can see that it is actually much more complex, but hopefully what you can see are those microtubules that have organized in such a way, and there too you could draw an imaginary line down the center of the chromosomes to indicate where that central axis is. So metaphase is characterized by the chromosomes aligning along the central axis. Anaphase, the phase that's in the center of your slide, is defined or associated with sister chromatids pulling apart from one another. So those microtubules, again, that are attached to the centromeres, pull apart the sister chromatids so that they move towards opposite ends of the cell. In telophase, the chromosomes begin to decondense or uncoil. You can begin to see the nuclear membrane forming, and you can begin to see where the cell will eventually divide in a cell division event. Now, mitosis, again, is a nuclear division process and follows what's happening with the genetic material in a nuclear division. If these phases that we just went through are followed by a process known as cytokinesis, that is just a partitioning of the cellular material and a complete cell division event, you then have true cell division taking place. So mitosis is not synonymous with cell division. Mitosis defines the nuclear division event. If mitosis is followed by cytokinesis, you then have cell division. Cytokinesis is the division or the partitioning of the cytoplasm with an eventual separation into two distinct cells. Now, animal cells and plant cells do this a little bit differently. So on the left-hand side, we're going to focus on animal cells. With animal cells, what happens is there is something known as a contractile ring that forms that allows for the eventual separation of the two cells. This contractile ring is made up of both actin and microtubules and the motor protein known as myosin. We highlighted this briefly when we talked about the cytoskeleton and motor proteins such as myosin and how they interact with actin. But more specifically, what you can see in both the electron micrograph image as well as the cartoon version is that essentially what you're having is a contraction that's occurring at the center of the cell to eventually pinch in and cause the two cells to separate from one another. In plant cells have a cell wall, and so there's a little bit of difference in the process of cytokinesis that occurs in plant cells. Because plant cells have a cell wall, what essentially happens that results in the formation of two daughter cells at the end of mitosis followed by cytokinesis is that vesicles that contain cell wall material are sent to the center of the cell that's going to be dividing. Eventually, these vesicles begin to fuse, and you have what's known as a cell plate form. And eventually, enough cell wall material is sent to the cell plate region of the cell to then essentially build a cell wall between the two cells, partitioning off the two cells in the plant cell. Let's go ahead and move on to talking about meiosis. In sexually reproducing organisms, meiosis is a special two-stage type of nuclear division followed by cell division, which will result in the production of gametes. And we're going to discuss some additional terms so that we can understand what is happening with meiosis. In genetics terms, the term haploid and diploid are oftentimes used to describe the chromosome composition of the cell. So a haploid cell is a cell or an organism that contains one complete set of genetic information represented by one complete set of chromosomes. A diploid cell is a cell or an organism that contains two complete sets of genetic information and two complete sets of chromosomes. Haploid chromosome number is typically designated by the lowercase letter n, and diploid chromosome number is indicated by 2 times the lowercase letter n. So if we talk about a typical human cell, in humans, almost every single cell of the body is in the diploid state, having 2 complete sets of genetic information represented by 2 complete sets of chromosomes. However, the gametes, either sperm or egg, need to be in the haploid state, containing 1 complete set of genetic information. So meiosis is a mechanism that allows for the production of haploid gametes from diploid cells of a typical organism. So going back to the human example, in humans, you may be familiar with the fact that humans have a total of 46 chromosomes. This is the diploid number. So 2n is equal to 46 in humans. Therefore, the haploid number is half of that, which is 23. So again, there is a direct relationship between the haploid state and the diploid state overall. Because I've mentioned the terms haploid and diploid, what I want to do is add one additional set of terms that's not listed on the slide, and that is a homologous pair of chromosomes. Actually, it is listed on the slide, but the definition isn't there. Homologous chromosomes are 2 copies of a particular chromosome in a diploid cell, one that came from the father of the individual and one that came from the mother of the individual. So in a diploid organism, you have 2 complete sets of chromosomes. 1 chromosome in a set came from your mother, and 1 chromosome in a set came from your father. Homologous chromosomes are chromosomes that are similar in size and shape and have the same genes located on them. Now, the specifics for what those genes determine can be different depending upon the genetic information present in your mother and your father. However, the exact genes that are located on the chromosomes in a homologous pair are going to be the same. In this slide, what we're going to look at is an overview of meiosis. Meiosis is oftentimes referred to as reduction division. It is divided into 2 phases, meiosis 1 and meiosis 2. Meiosis 1 is where the reduction takes place. There is a reduction in chromosome number that occurs during meiosis 1. And meiosis 2 represents what would be a normal cell division. So when we call or refer to meiosis as reduction division, meiosis 1 is the reduction, meiosis 2 is the division. And what you see on this slide again is the summary overall of what's happening. As we discussed at the beginning of mitosis, or at the beginning of the lecture, I mentioned that either for mitosis or meiosis, all of the genetic material has to be copied. So in the first part of this slide on the left-hand side, you can see interphase before meiosis takes place. You have a pair of homologous chromosomes that are present in that cell on the far left. And during the S phase of interphase, all of the genetic material is duplicated. So at the end of interphase, you have those characteristic X-like structures that represent the chromosomes present. And then from here, we can continue on into meiosis 1 and meiosis 2. Essentially, in the shortened, condensed form, in meiosis 1, homologous chromosomes separate from one another. In meiosis 2, sister chromatids separate from one another. And the next set of slides will look at that in more detail. On the far left, you have interphase, which again is the everyday life of the cell, but more specifically during the S phase is when the synthesis of DNA takes place. All of the genetic material is copied or duplicated or replicated during the S phase of interphase. So what we're going to do is enter into meiosis 1, and in meiosis 1 we can focus on prophase 1, This is where the chromosomes condense, the nuclear envelope begins to break down, and the mitotic spindle or microtubules begin to associate and form. More importantly, in prophase 1 of meiosis 1, we have a mechanism that is known as synapsis, and at synapsis, you have homologous pairs of chromosomes coming together. So, because in a diploid cell, you have 2 copies of every single chromosome, those 2 copies pair up during prophase 1 of meiosis 1. And this is unique to prophase 1 of meiosis 1. This does not happen during mitosis. So homologous chromosomes pair up, and more specifically, you can also have exchange of genetic information in homologous chromosomes. This is oftentimes referred to as crossing over, but more technically is referred to as genetic recombination. And we will talk about that in a little more detail when we enter into discussing genetics in our next unit of material. In metaphase 1, we see that those homologous pairs that have come together align along the central axis. So this is a little different than what we discussed for mitosis. How do those chromosomes move? Again, it's because the microtubules have attached to the chromosomes and they push and pull those chromosomes, those homologous pairs, towards the central axis of the cell. In anaphase 1, you have homologous pairs of chromosomes separating and pulling apart towards opposite ends of the cell. On the far left, we have telophase 1 followed by cytokinesis. Telophase 1, the chromosomes start to decondense, the nuclear membrane begins to reform, and essentially at the end of cytokinesis, we have 2 cells that are formed that are haploid. So again, this is the reduction portion of meiosis. We have gone from diploid cell at the beginning of the process to 2 haploid cells at the end of meiosis 1. Each one of those cells that has been produced by the end of meiosis 1 will continue on their own path into meiosis 2. Meiosis 2 is simply just like mitosis, and we can follow along those stages on this particular slide. So prophase 2, chromosomes condense, microtubules begin to organize. In metaphase 2, those chromosomes align along the central axis. How are they doing that? The microtubules have attached to the centromeres and push and pull those chromosomes along to the central axis. Anaphase 2, sister chromatids pull apart from one another by the microtubules pulling them apart, moving towards opposite ends of the cell. Telophase 2, followed by cytokinesis, chromosomes decondense, nuclear envelope reforms, and essentially those 2 cells separate from one another. So at the end of meiosis, you have 4 daughter cells that have been formed. Each one of those daughter cells are haploid. Now, as I mentioned at the beginning of discussing meiosis, unique feature or characteristic of meiosis is that those homologous pairs of chromosomes pair up, and more specifically, genetic exchange can occur between those homologous chromosomes. This is referred to simply as crossing over or genetic recombination. And in genetic recombination, the end result is the production of different gene combinations than those that were carried by the original parental chromosomes occur. If you work your way through the slide, you can see where that exchange of genetic information has taken place. Again, this only occurs in homologous pairs of chromosomes and is essentially reshuffling the genetic deck when gametes are produced so that genetic information, again, can be varied or diversified as gametes are being produced and the next generation is going to be produced. Now that we've gone through both of the mechanisms of nuclear division known as mitosis and meiosis, let's compare the 2 side by side. When we discuss meiosis, what we discussed at the tail end of this lecture was that you have a diploid cell that undergoes two distinct stages, meiosis 1 and meiosis 2, and at the end of the entire process, 4 daughter cells are formed. They are each haploid, and they are not genetically identical to the original parent cell. In mitosis, on the left-hand side, however, we are maintaining genetic composition. We're maintaining chromosome number. So you have a diploid cell that enters into mitosis. The end result is 2 daughter cells that are also both diploid. They are genetically identical to each other and genetically identical to the original parent. So the two mechanisms result in different compositions of chromosomes in the cells that result at the end of the process.